to non-VA entities about this research idea. This can be a university or an industry partner. However, a confidential disclosure agreement is needed before confidential information is disclosed. The STAR attorney will be able to help negotiate the CDA so that it is legally acceptable for a VA medical center to sign. Once the CDA is executed, the VA investigator and university industry partner will review the confidential information and determine whether they'd like to proceed with a collaboration. The STAR attorney can then help negotiate a cooperative research and development agreement or subaward for the study. This is typically done through the local nonprofit corporation's assistance. However, there are instances when the NPC would not be involved in the study. During the study, any invention that's conceived of or reduced to practice while performing the scope of work would be subject to intellectual property ownership language in the research agreement. Investigator would need to disclose the invention to tech transfer, and a DOR would be issued. Assuming the regulatory criteria to assert rights is met, VA's ownership interest would then be subject to the intellectual property language in the research agreement. Now, let's say a CRADA has VA retaining ownership in the invention that's created by a VA investigator, and the collaborator has an exclusive option to license this VA-owned technology. A DOR would be issued asserting rights in the invention, and then TTP would determine if a patent application be filed for the invention. Collaborator would then exercise their option to negotiate a license agreement with VA. Tech transfer would take lead in the patent prosecution matter, and STAR and TTP would negotiate the exclusive or non-exclusive license agreement. Another example that frequently happens is when a VA investigator has a research idea that is funded by VA and the investigator is solely a VA employee. The investigator conducts the research and, say, creates a new invention. The investigator would then disclose this invention to tech transfer. TTP would evaluate the disclosure and issue a recommendation to STAR. STAR would then issue determination of rights to the employee. Once VA has asserted this ownership right, tech transfer would be again taking lead on patent prosecution for the invention. And then tech transfer would work with other entities who are interested in licensing the technology. Once the company has been identified that's interested in licensing the technology, STAR and TTP work together to negotiate the patent license agreement. Now, I know I tossed a lot of information at you, but the key takeaway is STAR is involved from the very start to finish on research matters, whether it be the exchange of confidential information, the negotiation of research agreements, asserting ownership rights in a VA employee's invention, or licensing the invention to other entities. Now, finally, if you are a VA employee and have questions, please do not hesitate to reach out to your STAR attorney. STAR attorneys are assigned to specific geographic regions. Our assigned territories are broken down by states or specific cities. The local research office at your VA Medical Center or the local nonprofit corporation affiliated with your VA Medical Center can provide you with your STAR's point of contact information. Thank you, and I look forward to your questions.
So, questions for our panel with
Well, I'll start. I'll start by I'll just kind of kick it off and said, I really appreciate, even though these topics are extremely complex, I think you guys did a really great job with kind of educating us about what it is you do, what your offices do to help us navigate, like Rowan said, this minefield when it comes to disclosing your intellectual property to a company, what your position is as a VA employee, the sort of the ethical hurdle. So again, so many people, I mean, in science, it's all about open data and sharing. And of course, that I guess you can render your invention useless or not licensable if you do all of that without the appropriate legal advice. And I don't know if you guys, you or Matt or Chris, have examples where people have been just really disappointed and weren't able to go through to licensing because they just hadn't done things properly. You know, sort of like, I don't know if you have any kind of like sad stories to share with people, because I think I consider this session really sort of education for folks. You know, if they really want to work in this space and get their inventions to market. Over.
Well, I actually have one that I could build off of Dimitri because I was a records manager, and people don't realize it's not an exciting thing to do, but records matter, especially when if you're coming up to getting FDA clearance on things. You having a lot of that information documented and properly stored correctly can make the process smooth. I've been, scrambling, helping PIs when they didn't properly document properly and get all their ducks in a row for their initial screening with FDA, which is expensive and time consuming when you're not ready. And it delayed things by a large period of time. So, doc records isn't exciting, but it's important.
And so there's not so much I have an example of we got down the road of trying to license something and we had to call it off because of a ethics issue that couldn't be waived. By the time a license agreement reaches my desk, the tech transfer specialist has been working with it for a while, and we have kind of internal SOPs that identify, if a VA employee is going to be involved in the licensing, and they get sent to ethics for screening. So there's kind of steps to prevent it from getting too far down the road. Most of the time, deals fall apart because the potential licensee finds the terms not favorable and decides to walk away.
Yeah, I'll echo that, what Matthew said. We don't see a lot of bad results with licensing necessarily, but what we typically see are people who want to research their invention as part of VA research, but at the same time, as I mentioned in my talk, they have this outside company that they started to commercialize the research, and that's just a conflict that cannot be waived. So people just need to sort of think ahead. If I'm about to start a research project, do I have an interest in a company that is going to benefit from this? And if so, they need to talk to us, before they start going down the road of getting funding or getting the research approved, so that they don't invest all this energy into research that we ultimately say just can't happen.
Yeah, and Chris, sometimes people do start their own companies to access more money, so as you know, with invention, you're going to need more money, and that means putting it under the umbrella of a commercial entity or something. So that's why a lot of people end up going out. So I don't know if there's a way to do that from inside the government, and I'm not sure how many people have that experience of working within the government, developing something that VA clinicians especially, they develop something, they only want it to benefit VA, their heart's in the right place, but they just don't have, I guess that's where licensing comes in, where someone else can help them with, scaling scaling this product or something. So, and maybe I don't know if they would benefit. I guess they could benefit from the invention and the determination of rights, but that's why so many people leave. Is that they just need access to the kind of funding that you would get as a company. And I'm not speaking from any experience, and maybe Dr. Gerhardt can speak to this. It's a catch-22. Over.
Can I ask a question? When we submit the grant, we write it and we report all these like inventions, what we're going to do. Is it considered public, declaration, or it can? We had something very nice, but I've been told that you have presented in one of the meeting, and so it cannot be. It's too late. It cannot be processed for the invention, or what you
Yeah,
call like patenting and all that, like moving that area forward.
it depends on the information that was presented, and that's why I always tell as soon as you have the concept in your mind, get it into our system so that we can help guided along the way through the process, even if you're not thinking about commercialization, because we can, within the system, we can add to the records and we can help you along the way when you get an aha moment. But it's that helps. That's the first step to help kind of protect it and then guide you along the way with the documents that Matthew talked about, like, confidential disclosure agreement, or, a non-disclosure agreement, thinking about how you kind of build your conversations when you go out and present your papers or present your discussions with, and all it takes is discussion with one other person. People don't
Yes, sir.
realize that. So
So
that's,
one thing I've been told also, like you have like 12 months. So by the time we submit the grant and we get the money, we start, it's usually 9 to 10 men. And, so what is like the time frame we should do it?
well, if you're a VA employee, I would say disclose it to your tech transfer into Sophia. What you are probably thinking about is a provisional patent, and your idea may not be ready for that. That's our job, to help you kind of along the ways, working with our colleagues from OCG to get to that point where you have something, where we can do some background and find out a prior art. So don't think about, I only have 12 months. Think about, that's if that's what you have a provisional patent. But I understand, coming from academia of publish or perish, you kind of have to have a strategy on communication as well to make sure that,
Mhm. Thank you.
And I'll reiterate on that too, that the sooner you talk to the tech transfer specialist, the more their options are on the table and the more they're able to navigate and assist in this process. And, maybe it's something that is ready for a provisional application, and, to be able to file that, or maybe it's not quite there, but will be within the time frame they have to file something. Or maybe even, what you think is a disclosure under the grant, if there's submitted under confidentiality terms and it's not widely available or disseminated outside the application process. So there's caveats in all that, but the sooner you engage with the tech transfer office and the specialist and ask these questions, the better advice you'll get.
and to highlight, I'm sorry, but to highlight that most federal agencies have a tech transfer office, so it's not just, VA. So the Federal Lab Consortium, there are, you can find, DOI has it, DOD, Army, everybody has it in there. So, um, so you have lots of options, not just if you don't work for the VA, but work for another agency.
There's a question in the chat where someone asked if there's any advice about someone who wants to start a biotech company but still wants to work at VA. And the ethics answer to that is that there's no rule saying you can't start a company while at VA. And when I give ethics classes, I didn't do it today, but I usually draw a Venn diagram where you got your two circles. And if they intersect and you've got your VA duties here and your company here, that's where you have the conflict. So you can be a VA employee and also have a company. But if you come to us and say, I want my VA research to focus on this invention and the company's going to commercialize the invention, that's the problem. That's the conflict.
Dr. Gerhardt, you were nodding, so I didn't know if you had a question, or you can speak for your own experiences, because you've commercialized so much from your academic perspective.
Well, the key is communicate early and often. That has been pointed out, and I think the other thing to something that I often mention there. I think the world thinks that conflicts are a no-go. And virtually anything or even perception of conflict of interest as a whole can be worked out. So, in fact, our VPR office at University of Kentucky pretty much says periodically that there's no such thing as a conflict of interest that we can't find a solution for in the context of faculty pursuing, intellectual property and or other things. Like you said, if your duties cross, then we have to really, as been mentioned, we have to make sure that we watch for that. But really, the university, we set up these COI panels to independently look at what's going on. In my case, as I told you, I've had a company really based on academic results for over 25 years. And the story I sometimes don't tell people is that was all, actually you ask yourself, why did I make a company? I was encouraged by the National Science Foundation back in the 90s to form a company. And quite frankly, the program officer says, look, why don't you start a company so you can make money so I don't have to, pay for your grants. and so they looked upon it as a win-win scenario. And then whereas I think in academia sometimes they say, oh, we're a second-class citizen for having a company versus being your more stout academician. And the government really as a whole, I've found, no, they want you. That really was all born out from the 80s of what Bayh-Dole and the like, that they want you to strategically take advantage of tax dollars and with discovery. And if you invent something, they want that put back into our greater pool of not only technology, but growth of programs. And that's why I think if someone hasn't mentioned this, it's pretty much the case at agencies like NIH that there's more SBIR money available than is spent, usually on a yearly basis. And so it really encourages all of us to, make those transitions even with other companies. And so the two SBIRs that I currently have, I am the university PI for those companies. So, a certain amount of money comes to university, and then the other part goes to the company for the development of a product. So you get indirect costs for the work at the university. So it really can be a win-win scenario. So I encourage a lot of people. It's not, I think some people don't enter into the business development from your academic post because you think that it's going to be too much red tape and too many problems. But if you go to your intellectual property group early on, and even your chair often knows what's the first step. I have this great idea. I have a grant that I'm putting in that there may be an invention, and how you can file basically protect that intellectual property. And in fact, many of us file those things with our IP offices all the time. And so the message that some of us like to say is don't be afraid to go into that area, because we're all, whether you're clinical, basic science, or perhaps even more from a company perspective, I'm constantly working with people in corporations that quite frankly are worried about working with people in academia. So it goes both ways. So you break down those barriers, and there's really the tremendous thing that I found in working with corporations over the years is the sheer money in kind that also can be granted to help you in your research efforts using, naturally, if a company makes coat hangers, they're very willing to let you have some of their coat hangers because it's what they have and they can share that. And the coat hangers get pretty expensive when you get into biotechnology. I might say a few DBS stimulators and a few like implantable pumps that are FDA approved. We're talking some pretty heavy hardware. In fact, I was using one of these commercial available pumps at one time as an infusion pump because I had them, even though they were 10 times more expensive than an infusion pump, but they did such a great job. And so you have those opportunities too. So you don't always think about the money, but how can you advance your research and your translational program by making these ties with industry, and in fact your IP offices are really willing to help you with that side too. So I just encourage people to communicate and not be afraid.
I'm not sure if either Matt or Chris had a comment on, I mean, would that come under sort of a research agreement with the company? That is, I agree they're not sort of sharing money, but they're sharing equipment or in kind. Sometimes they may even pay for a person or something in your lab. is that something, Matt, maybe that would come through like a sort of a CRADA agreement?
Yeah, so and the agreements we do are under the Federal Technology Transfer Act, and under that you have the ability for your collaborator to provide funds or maybe just the device or drug. They could even potentially provide some personnel for some issues.
Okay.
And then we start getting scenarios where some of the funding they're providing can be used for additional personnel. The money comes through the local nonprofit, and so there's ways to set it up and structure it. But that's the big advantage is you can get equipment and supplies provided by the company that you're doing the study with.
Okay, great. And then, Chris, I don't know if, within VA, we do not have access to this SBIR funding. I know it's available through, like I said, NIH and other agencies. I was at Department of Commerce. They have that mechanism where, you would have this kind of shared role with a small company, and you're a university investigator working with a small company. Is that something that VA should think about? Sorry, Chris, I'm just putting you on the spot.
Well, I don't know much about our authority to do that or how that would really work with the ethics laws. But to comment on one thing that Dr. Gerhardt said, the universities are subject to different conflict rules than the federal government, and that's why I think why he said it's very easy to overcome a conflict on the university side, because it's all about what their policy says and what they're willing to tolerate. They're not subject to federal laws that say you can or cannot do a certain thing. So a lot of times investigators come from academia to VA, and they're kind of shocked with the strictness of the ethics laws. And our hands are tied by Congress in that regard. So I think that explains the difference between VA and the affiliates.
Arun, I see your question in the chat. Did you want to unmute and ask your question?
The few things are that, as Chris mentioned, like we are like intramural employees, like NIH, extramural employees are the university affiliates. So the rules there are difference between intramural versus extramural. So sometimes people, investigator, get grumbled about NIH can do it, why can't I do it? So simply what you are. The question I had was like, and also backing up on what Dr. Gerhardt said, not every investigator can make a royalty of over 150,000, which we as an option to an investigator can get it. So there is a time and place when you decide whether I need to start in a company. Usually the rationale should be that this is something company would not move it forward or would really need my expertise. So I think these are the discussions like lawyers and tech transfer officers should do it before an investigator say, I want to start up a company with everybody think they're going to make millions of dollars. And it'll be a really good idea to have that data and say, okay, 0.1% of all the VA licensee get more than 150,000. So you need to think before, is it worth the trouble you want to proceed on that path? And yes, if it is something you believe in and you believe nobody's going to do that, that is a good justification for you to go for it, even if there is no money. But they need to evaluate pros and cons rather than just grumble because I can't start, I don't want to do translation. I think there is enough royalties available to you to be in VA, do great science and do it. Or as Dr. Chris Pratt suggested, keep your sphere separate between what you do at university versus what you do at VA. And I think that would work. So like, I would really love to see this kind of a data out there so we can inform the investigator when we tell them, please go ahead and do some translation. And the argument you hear is, what's the point translating? I can't start anything. Thanks.
Yeah, the point I was going to make is from some of you know I have a long history actually in psychiatry, not only from the drug development, but also as faculty. And I'll tell you a tidbit about technology and starting companies. So in areas of Silicon Valley and chip manufacturers like in Texas and other places, there are actually psychiatrists that specialize in the type of depression and anxiety that results from individuals thinking they should start a company and make a lot of money and leave their academic or even corporation. And so it's good to keep in mind that many of us, it starts small and think low. And that if you're starting a company, the benefits of starting a company aren't just the monetary dollars, but don't think that you're going to make a lot of money overnight, unless I guess you're an Elon Musk or and I think he had slow times too, and so literally there are people that get kind of that dream, and so be very keen on the reason I really started my early biotech company was as a foundation to aid also in the things that I was interested in. So actually having, in my case, I have a company that actually provides supplies and materials, the things that I can use in my own laboratory and actually offset the R&D costs of things that I was already doing. So there was a lot of, ability there, but don't get, in fact, there's a world out there in psychiatry. We treat a lot of people with major depression that have thought that they're bankrupt because they had a great idea, and unfortunately, I've seen it in the pharmaceutical industry and the tech industry. There's just a lot of people out there, so keep that in mind. That I think the smarter thing for IP development is first to work with your office in many cases on basically patenting and potentially in the future licensing that technology, which is the big payoff for a lot of people because it rarely happens. But there are many reasons to start a company, and money isn't necessarily, ironically, the reason to start a company because it can be a dual-edged sword, as they say, painfully. I unfortunately cannot speak because of HIPAA-related issues about some of the people we treated years ago. So,
so thank you for sharing your insights.
And I wanted to follow
Sorry,
up
go
briefly
ahead.
on what
Yeah.
you're talking about. So one of the good advantages I like to tell investigators, when we're going through this process is, look, you're going to disclose the invention to VA, and let's say VA asserts rights. And then the tech transfer office, they're going to say, let's file a patent application on, these claims. And then say the patent gets granted or you're able to license it while it's still a patent application. There's, economy of scale there. You're not paying, you individually are not paying for the patent prosecution, which is very costly in itself. And the VA has tools in place to go forth and try and market that technology to potential licensees and bring in people who are, wanting to license it and bring it to market. And our goal is not necessarily a profit center. It is the primary goal is to get the technology out into the public space. And I think that's a very different mindset than private companies because it's about the dollar value. And yes, royalties are important and they help fund other things at VA, but that's not the only consideration. And the goal is to get the technology out there.
Thanks, Matt. You make a really good point there that VA people aren't in the business of making money, but hopefully bringing solutions to veterans, and we can't do it without involvement of companies. So again, thank you. I want to thank all the panel members for sharing, again, their expertise. Dr. Gayhart for jumping back into the discussion. I appreciate it. Chris, we didn't get to you. Did you have any commentary that you wanted to share, sort of from your perspective in the VA Ventures Lab and what you're trying to do there with biofabrication and again, dealing with patient data internal to VA. And of course, what we worry about is sharing that data across the enterprise, which is like the bigger problem, not just in Seattle, but what if we wanted to share that across the enterprise? So I don't know if you had any comments on that before we close out.
Yeah, I just wanted to pick up on something Matthew said.
Yeah.
He said they have SOPs, standard operating procedures, as things come into their office. So I think when you're handling patient data, I think that's pretty critical. to have, written documentation of how you handle things. And that just makes sure that everybody's on the same page. And then it's easy to reach out to your privacy officer, your security officer, your records manager, and make sure that they're all in concurrence with what you're doing. So I just wanted to emphasize the documentation part, not just on the records themselves, but on how you handle those things. For us, that's quite easy because that's part of our quality management system. We develop work instructions for everything that we're doing and procedures as well. So just want to emphasize that documentation part. And if there's any questions, happy to see if I can address them.
All right. Well, with that, I'm going to kind of close out the session. And Nicanor, are you going to join me at this point? But anyway, I want to thank all the speakers again for sharing their expertise and walking us through what we consider like really difficult questions. But as you can tell, our tech transfer team and our STARS team and OGC are here to help us navigate, like Ron said, that minefield. So again, thank you very much, and thank you, Chris, for stepping in for Dimitri and yeah. And what it takes to run something internal to VA with a quality management system and record keeping. So so with that, Nicanor, did you have any sort of closing remarks that you'd like to add?
First of all, I think it's a momentous accomplishment. The biofabrication community, it's well and alive. And I was impressed by a comparison with last year's symposium. One can see how we evolved in terms of focusing, in terms of even polishing the language and the concepts. Everything becomes more functional. It's a definitive increase in the quality of the participation and of the output. We cannot solve problems in one symposium, but there is one full year in between symposia. So please stay with us, come back with all other questions you might have. Please participate to our monthly engagement meetings and also with our mini symposia to supplement this huge information which is coming up. And thinking of on this curve of how things have evolved from last year to this year, please project it and be prepared for even more, even better quality of activity and consistency, hoping that this is exactly the mission which we try to fulfill by very existence of this community. In this regard, please don't forget to fill in the questionnaire which has been and a survey added to the meeting, because we cannot improve without your contribution and your participation. The feedback is truly essential. That is my first I would say reaction. Probably there will be much more coming up. By the way, we plan to produce a position paper which will summarize the discussions last year and this year in a sort of statement of intent and accomplishments, and to project in the near future what this community of science can do. Thank you.
And like Nicanor said, please fill out the survey, because we don't know how to do this better. We only have a certain perspective on the field, and are there other topics that we should cover or are being covered in other areas, and we can probably invite those speakers in. Again, this is an extremely complex area to navigate. So again, really appreciate bringing in outside speakers and experts to help inform us as we travel along this pathway, this translational pathway again. So appreciate all the folks who joined us today. And stay tuned. And please definitely come to our monthly meetings for more. Oh, Dr. Gerhardt. Dr. Gerhardt, are you clapping or raising your hand?
I was just clapping.
Okay. So with that, Nicanor, we said we would open up breakout rooms, but I don't I think what we'll do is just sort of remain. I think what we'll do is just remain in the main room. If anybody had any questions or suggestions, you know, and our speakers have given you their contact emails and things like that. And if you need that contact email, you know, kind of email us and we'll make sure to get that if you want to get in touch with, like, you know, discuss the ethics questions or your STAR team or something. So, so thank you very much, folks, for joining us today. And I hope you enjoyed it. I know I learned a lot every time.
So, sorry, so there's no breakout room?
Do you want a breakout room, Greta?
I would stay
Ravi,
on if there was.
Ravi says he wants a breakout room. Which breakout room do you want? We can have one on the you know, the non-invasive assessment with Greta.
Yes, I will go and open it right now.
Okay, okay.
But that probably
Oh, no
requires-
problem. I mean, if you think it's just a few of us because people are signing off, I guess. If you just want to be all in one big room, I'll just listen in.
Yeah, because yesterday we didn't have many in the breakout. I think it's been a long day. So, yeah.
Just stay a little bit longer.
Yeah, yeah. Well, we can ask her. Hi, Monica. Yeah. Monica was clapping.
I just wanted to pick Alex Chortos's brain a bit more because he said that he sees that there's a need to combine minds from you know, soft electronics folks, right? Sorry if I'm mispronouncing, right? Just wanted to see, as you know, I just really wish I could have your presentation to learn more from those papers as well. Can you explain more where exactly can we reach, you know, what field would be the best to merge with, that you see a clear gap as you were you know, preparing your presentation or and also doing your research? Where do you see an intersection that we don't currently have? I don't hear you.
Alex, we can see your mouth moving, but I don't know. You have lost your audio for some reason. Okay, that was you.
Yeah, I'm not sure why it was. You still can't hear me.
We can hear you now.
Oh, okay. Yeah, I guess I was just mentioning that, like, you know, I've been working with this person at Purdue on mechanobioreactors. And it seems like there's just a lot of, you know, transferable skills from what I was working on previously to working on this, you know, new topic of bioreactors. So that's kind of all I was pointing out. I wasn't, you know, mentioning a specific field or a specific person. But if you have particular, you know, needs or a particular problem that you're looking to solve in your bioreactor, I can maybe suggest some people who work on that topic. So I don't know, did you have like a particular type of topic that you're trying to solve?
I think I found that person. That person was you, because during our previous discussion, you said that you were also interested in incorporating the sensors in 3D, which you already do, I guess, right? And then having your sample as a sensor. I think that was one of the, instead of incorporating more electronics on top of designing in such a way that whatever you have is a sensor as well.
Yeah.
That was very attractive idea that you mentioned that I would like to explore more.
Oh, okay. Yeah. Yeah.
And then anything that would, because data is increasingly getting harder to handle, and then with the imaging as well. And someone pointed out that we don't necessarily have to see things all the time. So finding that balance of where exactly do we need the imaging and where do we not need imaging, and to minimize the amount of data that we store, which we never delete.
Yeah.
Yeah, that was one of the things that you also pointed out as a weakness in my our right OCP, because they're like very very large files to do it.
Yeah, that's definitely, I feel like we would need to kind of get some people together to figure that out, right? What actually needs to be measured.
And I think that's one of what actually needs to be measured is a big question for a lot of measurement. But yeah, thank you. I'll definitely reconnect.
I think we have a more specific example on how biosensors can be used in some biofabrication instance. It's the reason I connected with Alex, because we are developing a digital twin to a mandibular implant. And we have both the object and we have its computer model, which is finite element model type of system. And we wanted to convert it in a digital twin, for which we need to collect data about its biomechanical, actually mechanical properties. And in this regard, a pressure sensor, it's planned to be incorporated in the device to collect information about the pressure during mastication, and also possibly on the stiffness of the object in terms of bone growth inside of such a device. So that is one of the circumstances. I don't know if Brent Craven is still here. We asked him if he knows about concrete examples of digital twins on devices, and he said he's not aware, but we must bring up the fact that at least one project seems to be active in VA research, and that is specifically for the purpose of creating digital twins, which cannot be done without sensors for the feedback information.
I was going to ask Alex the question. I think it's the reverse question, which is not just the sensor, but maybe the stimulus. So maybe you can stimulate the tissue so that it will mechanically respond or electrically be, stimulated so that it will mature. So I was sort of thinking like, during the sort of the earliest stages of maturation, there may be a need to gently stimulate the new tissue so that it will mature, even if it's in a sort of mechanically active site. And again, sort of in the reverse, right, then determine the maturation based on the feedback from the biosensors. So kind of like this whole circuit, a whole circuit of information.
This is an excellent idea, and actually is required for the bone maturation to have mechanical stimuli. So such an actuator, as it is called, the device capable to act on something based on the information, it's could be part of such an system. Alex?
Yeah, I just wanted to mention that, people are doing a lot of mechanobiology studies and finding, that a lot of these parameters, like the magnitude of mechanical stimulation, the frequency of mechanical stimulation, electrical fields that apply, they're all kind of interdependent. Right. And so that's why we're really interested in these kind of like low cost, small scale mechanobioreactors with mechanical and electrical stimulation. We could potentially, just multiplex them and do, 1,000, conditions at a time to see what works the best. Because they're finding these effects are very kind of complex and interdependent.
Right. Yeah. And obviously the tissue responds to the mechanical load applied, right? As it does in the body, which is why we get this very fine architecture in our skeleton and things like that. Yeah. To resist load, for example. And Alex, you mentioned something about, did you say sort of a skin product you were looking at? And it was very, very at the very beginning of your talk. So was that like a wound healing product? You know, was it an artificial skin product that you mentioned?
I think, are you talking about the tissue stiffness monitoring device?
Yeah. So was
Yeah.
that like a wound, would that be used to sort of detect wound closure, or I
Well,
guess, yeah.
Yeah, I was kind of bringing that up just as an example of like, the electronic skin community has developed strategies for things like making scalable arrays of sensors that can measure things like stiffness. And so far, those, as far as I know, those technologies or strategies haven't been translated into the biofabrication community. And so that is one potential way, if you wanted to make something like an artificial heart, like a couple of the people were presenting about yesterday, you can't have an artificial heart that's immature. It would need to be mature.
Right.
And you would need to measure things like what are the stiffnesses and mechanical properties on something like that. So I was kind of just mentioning that there are these ideas and technology platforms in the electronic skin community that might be able to be useful within the biofabrication community.
All right, so maybe I misheard the skin part, meaning it's sort of a skin as it can be put on anything, as opposed to like just the surface of, human skin or something. Maybe that's what I was, that's what I was hearing. But basically very flat, very flexible biosensors that can be placed like on surfaces. Okay. All right, well, I don't know if we have any more questions for people.
I have a question.
Yeah.
I don't know if I'm gonna phrase it correctly, but yesterday we talked about skin, but it wasn't real. It was more like a patch because it didn't have the sweat glands, right? It didn't have the properties that we would think that our skin has. So for a person who doesn't have any sensory activity because of injury, let's say, if you put this artificial electronic skin, do you foresee that with the help of these electronics, you can send signals also to the brain so they can feel certain things?
I think you're talking to Alex and his haptics technology.
Yeah. So that
Alex?
was basically the topic of my PhD. Like, how do we actually make prosthetic skin? So skin that has the same electrical functions in terms of sensitivity as normal skin. The challenge there is that we were making those devices purely out of synthetic materials like rubber, right? So they wouldn't like actually eventually become integrated with the body. They're just kind of, something that you can put on something like a prosthetic device. And there's been enormous progress in that field in the last 5 years or so.
So possibly through the use of like nanotechnology, infused materials that provide a biofeedback kind of mechanism.
Yeah, certainly.
So instead of a rubber platform, it will be a biofabricated platform with the ability for you to sense through, right, to bring sensory information through your biologic film or something.
This brings up the other question, how the bioprinted skin, which is alive, it's similar to the normal skin. And we heard yesterday open recognition of limitations of this. There are still serious barriers towards the clinical application of bioprinted skin, not in short term. In short term, is going to be extremely beneficial. In long term, ability to integrate and convert into a fully functional organ, as the skin is, it's still a long way to go. So lack of blood vessels is one of the critical problems because through capillaries, those leukocytes with the reparatory function and immune function are continuously brought into the repairing skin. In this regard, it's remarkable that the 3C patch, in a sense, is similar to the granulation tissue. The granulation tissue is nothing more than that. It's fibrin, it's platelets, and it's leukocytes, which fills a hole. So 3D, 3C patch.
3C.
There are 3 cell types. Probably it's where or so comes from, or 3 layers. But that is a sort of artificial granulation tissue. I was surprised to see that that requires to be replaced every week. After a few days, it seems to lose its ability to continue to stimulate. Probably some improvement can be done there to convert that leukocyte layer in something more permanent and to more powerfully stimulate the tissue around in a deep skin wound. It won't be the epidermis only, except if it is superficial, but normally it could be a deeper layer up to even to the muscle or so. But what is remarkable is that in circulating mononuclear cells, there are progenitors for pretty much all tissues, including for skin and for muscle and for you name. And the potential to take advantage of that cell population and to help it differentiate in situ and then vastly improve the quality of the graft, of the skin graft. At that point, one can combine that 3C patch with the bioprinting skin, which will represent the top layers, the epidermis and dermis, which can be now done, but not the hypodermis, which is exactly that interface where most of this integration may take place. And to move things even further, the principle which is used in Biobond, and that is to use capillary fragments taken from another place and incorporated in the implant, could even further bring this construct to a higher level of natural composition and possibly ability to integrate. So it seems there are a few technological opportunities now which could very much improve the outcome of the technology, starting with bioprinting, but adding other layers of complexity, including the feedback in terms of function sensors, and eventually recreate the organ. So it's remarkable that discussion about skin took place here. The importance of it has been stressed enough, and to see now whom else we can aggregate in a skin biofabrication group to keep this momentum going and to take advantage and to help mature in clinical applications what is the goal of the activity.
Well, the 3C brought up to me, I was thinking about the patch and then thinking about Dr. Brittany Mears' diabetic soul implant. So figuring out what support mechanisms could probably further or help support that, because you don't want you want the patient to be somewhat active and mobile, right? So they don't lose other functions, but also thinking about how maybe that pairing could work together to improve the outcomes of that. I mean, for us, diabetic foot is an issue within VA. So I was thinking about, to me, that was like a natural match between 3C, and then Beth Ripley's group working here with the 3D printing, because you have a approved 3D printing a device and
Right.
C1, and then you have another clinical aspect that's approved. So trying to match that to see if there are improved outcomes or not.
We are putting together a mini symposium on skin biofabrication to come in the next few months. So please stay tuned and participate, because it's going to be really interesting.
Yeah, I think our development in skin is sort of distributed across the enterprise. Rowan, they don't all reside in R&D. Maybe there's some in the innovation ecosystem. So again, getting everybody on the same platform and kind of comparing notes would be really helpful. Yeah.
Exactly. Herding cats.
Yeah, yeah. But to me, the 3C patch reminded me of the earliest tissue engineering the body does. It's the fibrin clot. That's what the body does for wound healing. That is bio-inspired wound healing, which is the fibrin clot inside the body, which is sort of has a paracrine effect, right? So it just traps all the, you have your fibrin, and then it traps all the blood vessels and the cells, maybe not in such an organized way as in the 3C patch, but you don't introduce anything foreign. It's the patient's own samples. I don't think it's an expensive technique. I think maybe the vials might be disposable. That may be like the cost there. Yeah, and it was surprising that the skin doesn't integrate. Like you said, it was more like a bio bandage. It wasn't didn't appear. It was promoting wound healing, but it did not become part of the healed skin, at least based on what we heard. But I thought it was just really exciting to see that. I mean, the beauty is in its simplicity, at least. I'd like to say that. It was elegant.
Do you think we can collect the contact people from all the, let's say, skin departments, dermatology or so departments in VA, and directly connect and invite them for a discussion about the clinical needs on one side, and also to enact a bit of the discussion we had here to inform them, because many might not know about even the existence of such prep like 3C patch.
Well, that
Yeah.
I think inviting Joseph, because they are looking at, they've got models for about collecting clinician-oriented data, so using some of the systems that have been developed to bring that in, so that you could probably make a project quicker or more, get some more practical outcomes. So that's my 2 cents.
Did you already get connected with 4D BioCubed in Maryland?
Yeah.
Yeah,
we
because
had to speak
last time I spoke with them, they said that they are trying to incorporate the hair follicles as well.
Yeah, so we may have them at the mini symposium. We may just reactivate that, because we were supposed to have someone from the DoD burn research program, but they couldn't make it for our symposium day. So the idea would be to definitely make it not only about VA wounds, but also about burn, which is not seen a lot by VA, but there may be sort of complementarity in that space. So not go it alone. There are developments being made in both spaces that can be shared or transferred or something. Yeah.
And then Mark Ferrer from NCATS, who spoke in our workshop at NIST.
Yeah.
He's bioprinting skin. He's the specialist in that. Also local.
Yeah, what he spoke to us about his Nicanor, he spoke to us
Skin.
about organoids or something. As Mark Farrar, remember Mark Farrar, who spoke to in our symposium?
Yes, he did. Actually, he summarized his work on bioprinting for drug testing, is what they are doing there.
I'm sorry, what?
Glass? So constructs for drug testing.
Oh, I see.
Yeah,
I see. Yes,
yeah,
yes,
different,
yes.
different kind of.
And I think it was skin too, because somebody in his office was doing that. They have a paper on printing skin, and it's even vascularized constantly.
I was wondering, Kimberly, you talked about translation projects that you kick off after your interactions like this. How do you pick from all these great research? How does your office prioritize what to pursue?
Yeah, we definitely depend on our investigators to take this on. If this is something they want to take on within the purview of their lab, you know, and so if this is something, you know, they have either the expertise, the training, and the ability to work together, then a lot of it is about kind of bringing different expertise together. So you get like sort of, you know, kind of what we call parallel acceleration in multiple fields. Nicanor can speak about his constellation. Nicanor, you want to tell people about your constellation? I don't speak about other people's research, but Nicanor is involved in one of the vessel constellation projects with Dr. Murphy.
Yes, yes. We want to speed up the treatments for critical limb ischemia, and there is an opportunity to increase the viability of injected cells by providing in parallel with additional circulatory support. So it's a constellation because we are addressing the problem from different angles. It's not only muscle repair, but it's in parallel and equally important, the repair of the vascular system, which is damaged in critical limb ischemia. And how that is being done by using use of endothelial progenitor cells injected at the site before coming with mesenchymal stem cells or others, which indirectly will serve the same function, that is to stimulate angiogenesis, but not only. Also, incorporation of extracellular matrix, muscle-specific extracellular matrix at the site of injection, possibly pre-incorporating the cells into a hydrogel, because it's the environment where the cells will feel more comfortable. When we inject them right in the muscle, which is ischemic, it's dying, it's gangrenous, how you would expect to have a beneficial effect. So all this additional biomaterial and control cell behavior, it's now being enlisted to solve a old problem in a new way. And we consider this, it was the title of our project, New Regenerative Approaches for Treatment of Critical Limb Ischemia. Yes.
So it's like a multi-pronged
Thank you
approach. Yeah, it's a multi-pronged approach. Yeah. Yeah. And so we're very dependent, which is why skin has not really taken off, because I guess skin has always been pretty far in the translation spectrum. So, you know, we haven't got a lot of people working on that basic science, right? I mean, it's kind of moved out to industry and things like that. But it might be something we need to bring in, kind of clinically bring it in. Maybe not at the lower TRLs, but sort of in the mid-range TRLs or something. So, and making sure we work on the right models as well, you know, for skin. So that's why we didn't touch skin. Plus DoD is much better at skin than us, because they're all fix and burned people. You know?
I have one more question, since you're so close to the veteran population who often have conditions like traumatic events and PTSD. Is there any intersection between that area of science and regenerative medicine, what we are trying to do here, to also treat those kinds of conditions? Do you see any? Is there any work that is being done? Basically, I'm trying to see if there's any avenue that we can also contribute.
Like,
yeah.
So the brain, so what we're seeing is that folks, you know, so they're basically saying that there is systemic inflammation with people who have sort of a traumatic brain injury, and even in PTSD, they don't have the same healing response. And so impacts all your regenerative capabilities. So that people kind of studying what I call that axis, what like they call it like a brain bone axis, how conditions in the brain affect the skeletal system. And a lot of it has to do with circulating inflammatory cells and how that changes, natural wound healing in the body. So what you call systemic inflammation, so neural inflammation that kind of goes systemically.
Are you saying that you have researchers also like cell biologists who are also tackling that problem in your?
Yeah, they're tackling
your
what we call
agency?
that. Yeah, they call it like they call it the brain bone axis. So folks who are working sort of like between kind of like the Alzheimer's and the effect on the skeleton. There are people who work on what they call inflammaging, right? This axis and how that affects the skeletal system. Folks who work on like the effect of PTSD on the rest. So as much as you think it's a problem with the brain, it actually has these connections with the rest of the body. So but there are very, very few people who can do it. And the animal models for those, not the traumatic brain injury, but at least the PTSD in animal models is not something easy to set up. I mean, trying to give an animal PTSD might be it's questionable. I'm not going to go into the different models, but it's questionable whether you really do achieve something that you'd call clinically relevant. But they try to monitor based on like circulating cytokines and things like that. But
Thank you. That's very interesting.
Is the effect large enough to be measurable on the rest of the body? Yeah.
Yeah, I would question the ethical when designing the experiments, right? Aspect.
Yeah.
Thank you. I'm sorry, I'm dominating. Thanks.
No, it's sort of like animal behavioral studies, which is again a whole different specialty, and making sure you do it right and you don't kind of perturb the experiment and things like that, and whether the results will be meaningful. So they're very carefully controlled and have to be really well done and reproducible. So yeah.
I can imagine that.
Sorry?
I can imagine the IACUC being crazy. An IACUC for the approval for that.
Well, let's not talk about blowing up. No, I'm not gonna say. Like, the what is it? The VA blast core in Missouri at the School of Mining.
And this, I mean,
I'm sorry, Ravi.
no. I mean, this is really good. I mean, both points are good, and it's good to say that in the beginning than the after.
Yeah.
So there were a lot of issues with the animal protocols and animal thing, but as people said earlier, everything is resolvable if you start talking and thinking about it. So there was a lot of complexities. We still have fuel, but everything has been resolved. And with my personal experience, I'm using it, and there've been all this conflict, confusion, like how to transfer animal, how to do this, and all these animal issues. But everything got resolved at the end of the day. It took us 10 months, but it's it's resolved, and now things are moving forward. So and now it will set a record for the future. So it will not take this much time for other PI to do these similar studies in future.
Yeah. Wow. Yeah. Anytime you do those kind of studies, it's really hard. It's really, really hard.
But those are very important and very, critical studies from VA standpoint of view.
Yeah. So yes, Greta, if you want to know anything about the VA's blast core, then Rajiv and friends in Colombia, are doing those experiments. If you have, I don't know how many folks are doing stuff there. Rajiv is doing like the cornea work, right? You're doing cornea?
I'm doing cornea, especially with the special focus cornea, but rest of part of the eye as well. I've been looking at it. And the other group, neuroscience group, are looking at behavioral changes. So the whole idea is we do independently cornea, eye studies, and they independently, their behavioral studies has been done, and other neuroscience studies like changes in brain and all that.
Yeah.
And eye being the window of the brain, what the whole idea is, just do two studies independently and see if eye can be used to predict any risk, like who is going to be at a higher risk for PTSD and other issues.
Yeah.
So besides knowing what is going wrong in the eye, this has a more broader application.
Does that help, Greta?
Yeah. Can I ask, Rajiv, are you part of VA or I missed the university?
I am both. I'm part of the VA, and then I have a 6/8 in the VA appointment, and rest is the university. But I'm a tenured professor in the university.
Sorry, which one, so I can make a note?
University of Missouri. I'd be happy to
Missouri.
send you my information.
Thank you. Thank you.
Yeah. Yeah.
Well, I said VA, but it just sometimes when dealing with personal friends who have other type of traumatic events, I just wish there was the way we could just take it back or restore to the previous, I'm talking about like domestic violence and things like that. So of course, these kind of studies are too complicated, but I just wish that at some point I'm going to touch that in my career at NIST or elsewhere. I don't even know where to touch.
We should, and you should seriously consider it. If we find something, we can resolve major issues, major
All
issues.
right, I'll connect with you offline then.
Yeah, sure.
Thank you.
Yeah, it's funny how people talk about sort of psychological trauma as a true scarring in the brain. Like like your brain is scarred, the circuits are like become fibrotic. They're really hard to it's no longer plastic for that part of it. So that there's like a lot of parallels with what people say with like scarring and wound healing, right? It's a wound that never heals. Trauma as in psychological trauma. It's strange, that they don't really have good words for it, so they kind of use those kinds of things. And I guess the treatments have always been like, how do you kind of work through that scarring process or plasticity of that scar?
There was a patient story with the schizophrenia, and they said that the research there is so little that just imagine if you have a broken leg, and then when you go to a doctor and the doctor says, "So how are you feeling today? Tell me more about how are you feeling." So it's all about, there's no cure kind of a thing. And then that really triggered me. It was an audiobook, but then I just realized that there's a lot of conditions where we just talk about their feelings, but there's no solution. Like a trigger, people talk about triggers, and they went to counseling, and then they thought that, and then someone said something in a subway, and it triggered event 20 years ago. So is there a way to like reprogram the trigger to smile instead of cry? Like, I don't know. It's so interesting. But I'm a physicist, so I don't even know who to talk to about these things to see what.
Well, you're saying something. So I did this year. We had a Middleton winner who won the award for basically understanding the biological processes of schizophrenia. Basically, it turns out that the people with schizophrenia cannot determine when a sound is auto-generated, which means sort of like when they're talking, they don't realize it's them talking. And so they're actually having technically conversations with themselves, but they can't tell when and it turns out that it comes down to wiring and myelination of certain tracts. And when the schizophrenics were told that, they kind of felt better because they figured, well, it's not me, it's just my brain hasn't wired correctly. Now, have we got a treatment for that? Maybe not, but they've done a lot of again, this machine learning and AI, especially on the EEG, to determine, how well your insulation on certain parts, in certain wires in the brain. I mean, this is an area I barely understand, but it made sense, right? That if you don't have enough insulation, you get kind of these other signals crossing. So you can't tell, you can't tell if, or you're sort of talking, but you're talking to yourself and you don't realize it's yourself that's speaking. So it was just a very very insightful. Apparently, it came from songbirds. Apparently, the impetus for the human research came from these songbirds or something.
You never know.
Yeah.
Thank you. I'll look into it.
Yeah. Judy Ford. Judy Ford. Oh, sorry, Ron.
Judy Ford.
Yeah, I was going to say also CDC has done a lot of work on ACEs, so acute childhood experiences. So, you know, as a public health issue, and how you deal with the long-term traumatic experiences that children have had from these adverse childhood experiences. So,
I mean,
what was the beginning? You said something in the beginning. I didn't catch that.
CDC, the Center for Disease Control, has done a lot of work in ACEs, so
ACEs?
adverse childhood experiences.
I mean, the good thing is we start talking about it and start like thinking and investigating about it. So that's the first step to solve the problem.
Well, it's funny, like, you know, we thought we'll never solve the aging problem, and then we talk about rejuvenation therapy, and now we're sort of talking about behavioral problems, and maybe those are within our grasp to contemplate repairing those things. So
I mean, you know, you think about 25 years ago, anybody get cancer, that's like people start counting days. Now, you know, they can for sure live like 5, 10, and some even more than 25 years.
Yeah.
So it takes time, but now I mean, I'm excited.
So the diseases with a death sentence have become chronic conditions, and we can survive. Raji, we just have to solve the aging problem. That's all.
Yeah, that's right.
Before it's too late for some of us.
Invite David Sinclair next time.
This is, I mean, this is why we all talk about all the time wound healing. We just have to reverse the process, right? Just to have like a regeneration of more the tissue, restoration, and there will be no aging issues.
Thank you for staying.
Well, this is the part of the conference I kind of like, is sort of the, what do they call? I call it the corridor conversations. You know, sort of things kind of stimulate, and you get time to synthesize data and things like that. But I'm glad you joined us, Greta, for the 2 days.
Oh yeah.
There is something missing. A beer.
What? A beer.
We don't have a beer here.
This is the disadvantage of not in-person meeting. Greta, when is your next workshop? You're going to have another workshop?
We should talk about this. So I'm gonna reach out again to bring us all together, because with the manufacturing office, there was a lot of deadlines to produce some reports, and then so now I just finally, I think you saw the email that I posted the videos. Like even that was a task that like it was doable, but my brain was just not there. So that was the first thing, and the next thing is so I already made an outline and started writing the report, and then so I want to bring us together to make sure that we're all on the same page, that that's what's going to go in the report.
Okay.
And then from there we can think what to do next, because you know, this is a great space to meet people. But there's also idea of consortium at NIST that works well. It's a good model at NIST, where the keyword is pre-competitive space to discuss things that would solve not just for a problem, not just for one company, but for the whole field.
Yeah.
So I want to do it, but I need the buy-in from the stakeholders. So NIST would do it if I can tell them that so many companies or so many agencies think that it's a good idea. So that's what I want to touch and see. Do we need another workshop? Do we need to work a working group? Yeah. Should we work on a technical project, or should we do more administrative, like consortium?
Okay. All right. Well,
So that's my thinking.
Full time or not?
Well, yeah, 80% now I'm back. But now baby number 2 is coming, so I have to go on maternity leave again.
Congratulations, I think. Congratulations to you.
Thank you.
Now, were you thinking of going to George's meeting in July? Because you asked
That's the
about
thing.
it.
I had to cancel all the meetings because that's going to be my third
Upcoming.
trimester.
Oh, yeah. Coming up. Yeah. Wow.
I know you cannot see anything below,
Yeah,
below
no,
my
we,
chin.
well, we're glad that you can join us virtually anyway. That's the important
Yeah, that's
thing.
why I really like this, because you can join from anywhere. I
Right.
appreciate it, though. I'll reconnect again.
All right, then. Okay, guys, I'm going to log off at this time.
Bye.
And we'll be in touch by email. Okay. Thank you. I hope everybody had a
Appreciate
good
it. Bye.
time. Don't forget your survey, right?
Thank you. And I want to thank Kimberly for this, starting this very novel thing. I mean, it's one of the greatest thing, I think, which we all started learning, meeting with the right people and understanding the whole process, what it involves the translational research. Everyone says translation research, but how complex it is and how much, steps we need to know. And this platform is really very helpful.
Rajiv, we missed you when we had the cornea discussion.
I know. I was out of country at that time. So
I
sorry.
understand. That's okay. You missed a very, very good presentation. Again,
Yes.
it's
Yes,
very,
I
very
did.
complex. I mean, I didn't realize how complex the cornea is.
Yeah.
Thank you, Rajiv.
All
Appreciate
right.
it.
Thank you.
Have a good
Okay.
time. Bye.
Bye.
Nick and I will talk next week. Bye.
Bye.
Bye.